factors which affect the photosynthesis. We were discussing mainly sunlight. In that first property, which is the first property, that is light intensity. Uh, Light intensity determines the rate of photosynthesis. Yeah, when you see this graph, initially the rate of photosynthesis is low. Rate of photosynthesis is low initially because low light intensity. Low light intensity. With the increasing light intensity, the rate of photosynthesis also increases. So this part of the curve, what you are seeing here, A is limiting factor. Limiting factor. That limiting factor is light. So light intensity as the limiting factor here, what you are seeing. Half part of the B, half part of curve. This is also a limiting factor. So with the increasing intensity of the light, the rate of photosynthesis also increases. This what you are seeing, D. D. Rate of photosynthesis is maximum. Rate of photosynthesis is maximum. It has reached a peak. Okay, so it is maximum. At this light intensity, the rate of photosynthesis is maximum. It has elevated. It has elevated upwards. Okay, and see this part. Here, you are not seeing further increase in the rate of photosynthesis. Actually, the rate of photosynthesis can decline because in this other than light intensity, other factor can become limiting. Yeah, other factor can become limiting. Other factor can become limiting. Say for example, carbon dioxide can become the limiting factor. Even though high light intensity is there, but once it reaches the maximum state, This is the saturation point. It has reached saturation. Beyond that, even if you increase that parameter like light intensity, process will not increase, it will decrease because at this point, other parameters or other external factors can become limiting factor for the rate of the photosynthesis. So, C represents these all letters, regions represent these different what we call parameters that you not remember. Okay, see, other than the light intensity, other factor can become limiting. Therefore, photosynthesis will not increase, but it will decrease. Okay, so that is what represented in this scene. So this is the graph showing the rate of photosynthesis influenced by light intensity. As I mentioned yesterday, if light intensity is high, well, high light intensity it leads to photo-oxidation of chlorophyll molecule, photo-ionization, photo-oxidation of chlorophyll molecule. Okay. If the chlorophyll molecule gets photo-oxidized, further, if it is continued, light intensity continued to be exposed on the leaves, that can lead to destruction of chlorophyll molecule, including that photosynthesizing cell, that is mesophyll cell, including that photosynthesizing cell or mesophyll cell, that is, you call it as solarization. So this effects will be there of high light intensity. That's why high light intensity beyond the permissible level is not advisable for the rate of photosynthesis. It is represented as E. So here, the rate of photosynthesis has reached to saturation at this point. Beyond that, you can see constant rate of photosynthesis if conditions are favorable. Otherwise, the rate can decline because other factors other than the light intensity can become limiting. So this is about light intensity. At low light intensity, rate of photosynthesis will be low. Along with increase in light intensity, rate of photosynthesis also increases. That will keep it in mind up to saturation point. Beyond that, there will be decrease because of this photooxidation or because of solar radiation, whatever it is. Now moving on to second property of light, we have that is light quality. Light quality. So light quality refers to color and wavelength of light. Color and wavelength of light. So the light which is used for photosynthesis is mainly visible light. What we say, visible spectrum. Visible spectrum of electromagnetic radiations emitted from the sun. Solar electromagnetic radiation emitted from the sun. So which light supports photosynthesis, which light support the life on Earth, mainly this visible light or the visible spectrum of solar electromagnetic radiation. So this range from 380 nanometer to 760 nanometer or 3800 angstrom to 7,600 angstrom. This is the range of visible spectrum. So this visible spectrum, it has what you call it, 7 colors. 7 colors are there. So when the visible light falls on the leaves, on the plants, which color, which wavelength of light is more effective in enhancing, in increasing the rate of photosynthesis? Rate of photosynthesis. See, when sun emits the radiations, most of the radiations, these can be reflected from the outer atmosphere. Some of the harmful radiations are absorbed by the gases which are present in the stratosphere, the ozone layer. Is it or not? It absorbs harmful UV radiations emitted from the sun. Some of the radiations which reach the earth from the earth's surface, these are reflected back. Some of the radiations are absorbed by the earth's surface and that becomes infrared radiation, deep radiations. Okay. So the radiations which are utilized for photosynthesis, we usually refer it as PAR, photosynthetically active radiation. Photosynthetically active radiation. The incident light which falls on the earth, which can be used for photosynthesis, that constitute around 50% of the incident light. 50% of the incident light can be PAR. It is reaching the earth's surface. Light radiations fall on the earth. But the entire total radiations cannot be used by the plants, by the leaves to carry out photosynthesis. So actually we have 1 to 2% of the incident light falling on the leaves is actually used in photosynthesis. Hardly 1 to 2% of the incident light on the leaves, on the plants, on the green parts of the plant, that can be used for photosynthesis, that is trapped, that radiant energy is trapped and it is converted into chemical energy. Okay, so hardly 1%, 1 to 2% range. Actual incident radiations of solar electromagnetic spectrum or radiation utilized by the plants in photosynthesis. So these different color and wavelength of light, see, these are having shorter wavelength. Okay, these are having the longer wavelength. Shorter wavelength, higher penetrative. Longer wavelength, less penetrative. Is it or not? Longer wavelength means frequency will be less. Shorter wavelength means frequency will be more. That's why they are more penetrating. Okay. So out of these different color of the visible spectrum, which can increase the rate of photosynthesis. Regarding that, already we have learned what we have learned: absorption spectrum and action spectrum. We have already learned, right? We have learned about absorption spectrum. The graphic representation of different wavelengths of light absorbed by photosynthetic pigments during photosynthesis is known as absorption spectrum. Graphic representation of different wavelengths of light utilized by the plants during photosynthesis, that is absorption spectrum. Then we learned even action spectrum. We learned even action spectrum. T.W. Engelman, he gave the first action spectrum. Out of the 7 color light, which light is more effective in increasing the rate of photosynthesis? We measure increase in the rate of photosynthesis by measuring the number of oxygen bubbles released per minute. We cannot see whether the rate of photosynthesis is higher or lower or medium. just by seeing the plant from external view. So we have to perform the experiment on hydrilla plant. It is a submerged aquatic plant where we count the number of the oxygen bubbles released from that stem cutting from the branch cutting. Okay, so based on that we say rate of photosynthesis is high. So we measure the rate of photosynthesis usually by counting the number of the oxygen bubbles. So out of these 7 color light, which light is more effective in increasing the rate of photosynthesis as per the action spectrum what we have learned already. So that is mainly we have first red. Highest rate of photosynthesis is in red. It is followed by blue. First red, then it is followed by then the least lowest rate of photosynthesis is there in green. The surface of the leaves appear green because they reflect green light. You should not tell because of chlorophyll. Of course, chlorophyll will be there. Okay. So the leaf surface will reflect the green light. That's why it appears green. So highest rate of photosynthesis in red light. Red light of the visual spectrum. That remember. Then next in blue light. Okay, least lowest rate of photosynthesis in green. So this is what we call action spectrum. Okay, the graphic representation of effect of different wavelengths of light on the rate of photosynthesis. Already have written at that time only. The graphic representation of effect of different wavelengths of light on the rate of photosynthesis is known as action spectrum. Okay, so we have learned as action spectrum. Here, wavelength of light. Okay, so 400, 500, 600, 700 nanometer wavelength of light are represented. Right here, rate of photosynthesis. So what we got? We got 2 peaks when we represented that graph. So that peak, one like this, and another one will be like this. So here you have the peak that comprise the blue color, blue wavelength of light. And here you have a peak. 2 peaks you can see. But it comes, this color and wavelength comes in the range of red. This is what we say action spectrum. Graphic representation of effect of different wavelength of light on the rate of photosynthesis is referred as action spectrum. First it was explained by P.W. Engler. That's right. So among these different colored light in the visual spectrum, these are very effective in increasing the rate of photosynthesis. That's right. Now, in this regard, we have to learn 2 effects. Very important, not even in NCRT. See, if red light is more, it favorable for carbohydrate synthesis. It increases carbohydrate synthesis. Suppose if blue light is more, it favors increased protein synthesis. Protein synthesis. Remember, red light more means it favors rapid or rise in carbohydrate synthesis, sugar synthesis. If blue light is more, it can increase protein synthesis. Basically, carbohydrates are produced in photosynthesis. Sometimes even other substrates like fats and proteins can also be synthesized. Red drum effect. Red drum effect. Another one we have immersion enhancement effect. These two effects we have written. Immerse along with ladies, mainly immerse. They were doing research on chlorella, a unicellular green alga. Unicellular green alga. And this chlorella culture, okay, this is chlorella culture. This chlorella culture was exposed to red light. Only one color, only one kind of wavelength of light. This, when we expose to only one kind of light, that is monochromatic light. Monochromatic light. This monochromatic light, red light, of more than 680 nanometer wavelength. More than 680 nanometer wavelength, red light. Monochromatic light, single color light was exposed onto the culture of this chlorella. What they observed, there was sudden decrease or drop in the rate of photosynthesis. Sudden decrease in the rate of photosynthesis in a plant when it was exposed to single or monochromatic red light of more than 680 nanometer. So there was a decrease in the rate of photosynthesis as observed by Emerson and Lewis. This they call it an red drum effect. Called it a red drum effect. We say usually red light can enhance the photosynthesis, but the specific wavelength of red light is important. It is far red, more than 680 nanometer is far red wavelength of light. So when far red wavelength of light was exposed onto the culture cells of the chlorella, rather than increasing the rate of photosynthesis, there was sudden decrease in the rate of photosynthesis. This is referred as red drum effect. Now, this chlorella culture, unicellular green alga, this chlorella culture, now they exposed it to dichromatic light. Dichromatic light. Dichromatic light means two colored light. Two color. This time, not only they used red, they used blue. They used blue. So this time the culture of the chlorella was exposed to red light. Okay, red light of around 650 nanometer. It is not far red, 650 nanometer. In combination with blue light of around 440 nanometer. When the combination of red light and blue light was exposed onto the chlorella culture, they were surprised now. When combination of red and blue light was exposed, was incident on the cultural cells of the chlorella, now there is sudden increase in the rate of photosynthesis. There was drastic increase in the rate of photosynthesis. This is referred as Emerson enhancement effect. So instead of providing monochromatic light, rate of photosynthesis will be more in dichromatic light. So one is of red light of optimum range, not far red, and another one is of shorter blue light. When it is exposed onto the chlorella culture, it increased the rate of photosynthesis. This is referred as Emerson enhancement effect. So this is recording the color and wavelength of light that can influence the rate of photosynthesis. So this is light quality. So light quality is very important. Thinking that only red light can enhance the rate of photosynthesis, if you provide more red light to the plants, especially to the crop plants, then there will not be increase, but there will be decrease. So better for a plant to the leaves, we can give dichromatic light. So now, the third property of light, that is light duration. Light duration. See, usually long hours of light duration can increase the rate of photosynthesis. Usually, light duration of long hours can increase the rate of photosynthesis. Depends on the requirement of light for growth, development, for flowering. It depends on the plant. We have short day plant, we have long day plant, we have day neutral plant. Different group of plants are there based on the requirement of photo period. But usually, for the photosynthesis purpose, usually say around 10 to 12 hours of photo period or light if you provide, that can increase the rate we usually set. Light duration, whatever the rate, usually that will not much influence. That will not much influence, but light should be there. Okay, when light is there, as you know that light dependent reactions occur. Light dependent photochemical reactions occur. Light reactions occur. If the light reactions occur, there will be liberation of oxygen. There will be synthesis of assimilatory powers like ATP and NADPH. So enough quantity or enough amount of these assimilatory powers will be produced, and these will be there in the chloroplast of the mesophyll cells. Suppose if you provide only continuous light, thinking that the rate of photosynthesis increase, if you provide continuously light, only light reactions can occur. And when enough number of ATP and NADPH are stored, further there is no addition. Enough, the cell has already enough number of ATP and NADPH. These have not been utilized because they have provided continuously light. So there should be small duration of even dark phase is also very useful. Dark phase means no need of light. No need of light. So when you provide the dark phase, in that case, which reactions occur? Dark reactions can. Dark reactions can. They are light independent reactions. They don't need light. So if dark reaction occurs, they make use of ATP and NADPH assimilatory power for the carbon dioxide fixation and they synthesize sugar. So that's why usually there should not be continuous sunlight. Light duration should be interrupted with dark phase, absence of light, so that dark reactions can occur, Kelvin cycle can occur. They make use of assimilatory powers and fix the carbon dioxide to synthesize sugars or carbohydrate. That's why there should be light duration, in between there should be dark phase also. Hence, dark reaction should proceed. Suppose if you provide only dark reaction, only darkness, if you provide only darkness, no light, dark reactions will continue up to some time until all the assimilatory powers are used up. Thinking that if you provide dark reaction, it favor what you call dark reactions or Kelvin cycle in dark phase. If you continuously provide, then of course Kelvin cycle can occur. But Kelvin cycle are dependent on the products of light reaction, these products. So how long these products are stored or available in the cell, until that dark reactions can continue or proceed. One stage will come, one point will come, no more ATP and NADPH are available. Hence, there is no fixation of carbon dioxide. Hence, there is decrease in the rate of photosynthesis. That's why light duration should be intermittent, usually. Light of say 5 to 6 hours, followed by at least half an hour, 1 hour dark phase, so that dark reaction can proceed. Dark reaction can occur during daytime, night time, but they don't use light independent reaction. Right? So this is about the light duration. So these are the three properties of light. Next external factor now, second one, carbon dioxide. It is a major factor influencing the rate of photosynthesis. It is one of the major factor influencing the rate of photosynthesis. So basically the carbon dioxide which is there, it is there in the air. This is the external source of carbon dioxide used by plants. This external source is important. That is determined by the availability of carbon dioxide in the air. Even plants can use internal source also. Internal source of carbon dioxide. Internal source of carbon dioxide is coming mainly from respiration process. Whenever respiration occurs, a complex organic food is broken down into simple molecules like carbon dioxide and water with the release of energy. So this is an internal source of carbon dioxide. When it is liberated from respiration, can be utilized by the mesophyll cells of the leaves to carry out photosynthesis. Otherwise, mainly plants are dependent on external carbon dioxide availability in the air. So this carbon dioxide concentration in the air, 0.03% to 0.04%. 0.03 to 0.04%. Okay. So at low level of carbon dioxide concentration, photosynthesis will be low. Photosynthesis is low, remember. At high light intensity, C3 plants, C4 plants can respond in different way to carbon dioxide concentration. Different way to carbon dioxide concentration. at high light intensity, C3 plants are not much well adopted. Tropical and subtropical regions where high light intensity can be there, that can increase high temperature, that can increase the temperature. So C4 plants can respond better as compared to C3 plant at high light intensity. Okay, see, at low light intensity, even if high carbon dioxide is there, at low light intensity, even if high carbon dioxide is there, C3 plants and C4 plants do not respond properly. At the low light intensity. Even if high carbon dioxide concentration is there, C3 plant and C4 plants do not respond properly. With the increase in carbon dioxide concentration, photosynthetic rate increases more in C3 plants as compared to C4 plants. Because in C4 plants, that carboxylase is catalyzing the carbon dioxide fixation in the mesophyll cell. And another fixation which is there, that is carbon dioxide fixation in the bundle sheet cell by Rubisco, that carbon dioxide concentration will be always maintained because that carbon dioxide is coming from decarboxylation of organic acid. The carbon dioxide per Kelvin cycle in C4 plant is not coming from surrounding air. That remember. So usually, if you increase the carbon dioxide concentration, the rate of photosynthesis can be high in C3 plant. They respond in proper way because of increased carbon dioxide concentration. So up to say 0.05% increase, rate of photosynthesis can increase. Okay, up to 0.05%. Suppose if we increase the carbon dioxide concentration rate under laboratory condition and experimental research, up to 1% increase of carbon dioxide has shown positive result in increasing the rate of photosynthesis. In the open environmental conditions, CO2 concentration, if it is increased up to 0.05%, C3 plants have responded better and they have shown high rate of photosynthesis. But beyond that, if CO2 level is increased, this carbon dioxide can act as anti-transparent. It acted anti-transparent. That means higher carbon dioxide concentration can bring the closure of stomata. If stomata are closed, stomata are present in the epidermis of the leaves. They are the gateways of transpiration. Loss of water in vapor form from the green aerial parts of the plant is called transpiration. Transpiration is useful in certain aspects. So if stomata are the minute openings, if they are closed because of high carbon dioxide concentration, then there will not be exchange of gases between the leaves, between the mesophyll cells of the leaves and surrounding environment, if stomata are closed. So thereby, photosynthetic rate decreases. For photosynthesis, surrounding carbon dioxide should diffuse into the mesophyll cells. After photosynthesis, oxygen which is released as a byproduct that should diffuse from mesophyll cells of the leaves to surrounding air. So by simple diffusion from higher to lower concentration, there will be exchange of gases between the mesophyll cells of the leaves and surrounding environment. So if the stomata is closed, such exchange of gases can be prevented. Thereby, carbon dioxide necessary for photosynthesis will not diffuse into the leaves. Hence, photosynthetic rate decreases. Okay. The present concentration of the carbon dioxide which is there in the air, this present concentration of the carbon dioxide is limiting for C3 plants. That's why if you raise the carbon dioxide concentration in C3 plants, the rate of photosynthesis also will increase. The present carbon dioxide concentration, which is that, it is optimum normal range. Optimum is normal as per the requirement. Okay, optimum for C4 plants. It is optimum for C4 plants. It is limiting for C3 plants. Present carbon dioxide concentration. If we increase the CO2 concentration, C3 plants respond in a better way than C4. Carbon dioxide compensation point. Yesterday I have given light compensation point. Is it a light compensation point? Like that, carbon dioxide compensation point. The stage of carbon dioxide concentration at which there is no exchange of gases between the plant and the surrounding environment. Stage or concentration of carbon dioxide at which there is no exchange of gases between plant and environment means at this point rate of photosynthesis usually will become equal to the rate of respiration, equal to the rate of respiration. So carbon dioxide compensation point, at that point what happens is the rate of photosynthesis will be equal to the rate of respiration. So in photosynthesis what is released? Oxygen is released. Oxygen is essential for respiration purpose. After respiration, what is released? Carbon dioxide is released. Carbon dioxide is necessary for photosynthesis. So the concentration of the carbon dioxide, the stage of carbon dioxide concentration at which there is no net exchange of gases between the plant and surrounding environment. At this stage, photosynthetic rate will be equal to respiration rate, that is carbon dioxide compensation point. So carbon dioxide compensation point in the case of C4 plants is low, that is 0 to 10 ppm. For C3 plants, it is high, it is 25 to 100 ppm. Carbon dioxide compensation point for C4 plant 0 to 10 ppm. C3 plant 25 to 100 ppm. See, when you rise the carbon dioxide concentration up to certain level, rate of photosynthesis also increase. Beyond the permissible level, it decrease I thought because CO2 acts as anti-transparent, which brings the closure of the stomata, thereby prevents the exchange of gases. Okay, so up to certain level, the rate of photosynthesis can increase. So that's what we can say up to saturation point. Up to a saturation point, if you raise CO2 level, definitely the rate of photosynthesis will increase. Beyond the saturation point, it will not decrease. So carbon dioxide saturation point for C4 plant is lower. Carbon dioxide saturation for C3 plant is higher. C4 plants, carbon dioxide saturation. It is 360 microliter per liter. It is also written as 360 ppm. ppm is parts per million. Then C3 plants carbon dioxide saturation. It is more than 450 microliter per liter, or it is more than 450 ppm. Here, requirement of carbon dioxide is greater in C3 plant as compared to C4 plant. That's why what I told, the present carbon dioxide concentration is limiting for C3 plant. It is optimum for C4 plant. Okay. See, we grow certain crops in what I call greenhouse. Greenhouse. Greenhouse crops, what we call. Greenhouse means a big glass house will be there. Okay. So under that we cultivate some crops, some plants we grow, ornamental plant, horticulture plant, some crop plants we grow. Okay. So here in greenhouse, crop like tomato, bell pepper, these can be grown, these can be cultivated. So in the greenhouse, what happens? This is the greenhouse, there is glass house. Here we have kept the growth of the plant. Light will be falling. Light can pass through the transparent glass. These radiations reach the ground surface within the glass house. The ground surface get heated. Okay. In turn, the ground surface emits heat radiations. These heat radiations will not escape from the glass house. So warm temperature is maintained here. Warm temperature is maintained. That is the operation of greenhouse. Right? So here, these are referred as greenhouse crops. Greenhouse crop. So when you cultivate the greenhouse crops with the increase in carbon dioxide concentration, photosynthetic yield has been improved in the greenhouse crops. Photosynthetic yield has been improved in the greenhouse crop. That's why some of the crops are grown in greenhouse to enhance the agriculture production or productivity. Definitely. Okay. So if carbon dioxide is there, carbon dioxide is a heat trapping gas. So if CO2 is there, the radiations which are reflected from the ground surface, they will be retained within the glass house, not allowing to escape. Thereby, warm temperature that is required for the growth and development of the plant is maintained. And this carbon dioxide can be utilized for photosynthesis purpose. So, increased carbon dioxide concentration can increase the rate of photosynthesis in greenhouse crops, especially the C3 plants or that, right? So this is about what we call the carbon dioxide concentration. Okay, so usually with CO2 level increase, photosynthesis increases up to certain point, that is up to saturation point. Beyond that, photosynthesis will not increase. Experimental conditions different, research conditions different. It has shown nearly doubling the rate of photosynthesis by increasing CO2 concentration. Okay, this CO2, if it is high concentrated in the surrounding of the plant, it can be toxic to the plant. It can be toxic to the plant. It can act as anti-transparent, break the close of the stomach. Then the next technical factor we have temperature. Remember Carbon dioxide compensation point for C3 plant, C4 plant, carbon dioxide saturation for C4 and C3. These values are okay. Next, temperature. Temperature mainly influence temperature mainly influence biochemical reactions. Biochemical reactions, nothing but dark reactions. C3. As we know that in C3 cycle, each step is catalyzed by enzyme. Enzymes usually they are thermosensitive. They are sensitive to high temperature. If high temperature is there, enzyme get inactivated, denaturation of enzyme. If it get inactivated, the step, the reaction which they are catalyzing, that reaction will be checked, will be inhibited, or it becomes slow. That indirectly affect the rate of photosynthesis. Light can influence even photochemical reaction also, but effect of this effect of temperature on photochemical reaction, sunlight only will control the availability of light intensity and temperature. Every number. Okay, so this photochemical reaction is less affected by temperature because in light reactions there is no much involvement of many enzymes. No much involvement of many enzymes there. That's why photochemical reactions or light reactions, heat reactions are less influenced or less affected by the light, the temperature as compared to the biochemical reaction, dark reactions. Okay, each reaction step is being catalyzed by biocatalyst, that is enzyme, which are usually sensitive to high temperature. They get inactivated or denatured. That's why the reaction which they are catalyzing, that reaction will not proceed, or it is inhibited, or it is blocked. Hence, overall, it can affect the rate of photosynthesis. Optimum temperature, optimum temperature for C3 plants. Optimum temperature for C3 plants, it can be 20 to 25 degrees centigrade. This is optimum temperature. For C3 plant, it is 20 to 25 degrees centigrade. Whereas for C4 plants, optimum temperature for C4 plants, it is 20 to 45 degrees centigrade. High. It is high because the C4 plants where they will be growing tropical and subtropical region. Okay, and this pep carboxylase enzyme can work even at high temperature. Rubisco cannot work at high temperature. Rubisco enzyme, which is involved in carboxylation step in C3 cycle, get inactivated if there is rise in temperature. Range will be there, limited range will be there. Up to that, in C3 plants also, with the increase in temperature, rate of photosynthesis will increase. That we should remember. Rate of photosynthesis will increase with increase in temperature, but up to certain range. In C3 plant, it is usually up to 35 degrees centigrade. Up to 35 degrees. In C4 plant, up to 40-45 degrees centigrade, these can perform photosynthesis even at higher rate. That is because peptidase enzyme is tolerant to high temperature, whereas rubisco is sensitive to high temperature. If it goes beyond 35 or beyond 40 degrees, Rubisco enzyme get inactivated, that dark reaction, carboxylations have been ceased. No more synthesis of organic sugars. So that's why C4 plants optimum temperature is 30 to 45 degrees. Different plants are adapted to carry out photosynthesis at different temperature range. There are some plants, usually gymnosperms, which are growing in cold regions of the world, Himalaya, tundra region. Okay, temperature is less, 0, sometimes, especially in winter, -degree Celsius. Okay, so such coniferous trees, gymnosperms, they are usually capable of performing photosynthesis in the range of 0 to 5 degree. Sometimes even certain gymnosperms can carry out photosynthesis at -35 degrees Celsius. Certain gymnosperm coniferous trees can perform photosynthesis under -degree Celsius. Now, certain plants like xenophytes, cactus, which usually grow in desert conditions. In desert conditions, summer temperature will exceed high. Summer temperature can reach up to 60 degree in deserts. So, Papuncia, cactus, these are xerophytes, they can perform photosynthesis even at 50 to 55 degrees centigrade. Remember. So, the plants which are living in different habitat are well adapted to carry out photosynthesis at different temperature. But this is normal range which I have given. Now, there are spring algae, hot spring algae, hot spring algae. With these algae grow in hot spring. In hot spring, temperature will be more. Okay, they can perform photosynthesis at 70 to 72 degrees centigrade or more than 70 degrees centigrade. These can perform the photosynthesis. Hot spring living algae more than 70 degrees. These normal plants cannot perform at this temperature. Okay, normal plants are not adapted to perform at low temperature. And usually, what happens is at low temperature, enzymes will be least active. Enzymes will be least active. Usually, rate of the process or reaction will be very low. But due to specific adaptation of these plants living in different habitat, they are having such peculiar characters. Because of that, they can perform even photosynthesis in a different range of temperature. Okay, so temperature influence more on dark reactions than light reactions. As the dark reactions are being catalyzed by enzymes. That remember. Okay, so usually with the increase in temperature, photosynthesis also increase, but up to certain limit. Beyond that, inactivation of enzyme or denaturation of enzymes is there. That is how the rate can be checked or inhibited. So this is about temperature. Remember. Then the external factor, as per exact last external factor, that is water. 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 See, water is raw material, you know that. It is a raw material used in photosynthesis along with carbon dioxide. Water is the source of oxygen, water is the source of proton and electron. Okay. Usually, water cannot be a limiting factor. Usually, water can't be a limiting factor because usually 1% of the water absorbed from by the plant. Okay, whatever the amount of water being absorbed by the plant, 1% of the absorbed water, okay, will be used for metabolic activities like photosynthesis. Plants can make use of hardly 1% of water for photosynthesis. That's why usually water cannot be limiting factor. But water is essential. It is a raw material for photosynthesis. This water maintains turbidity of the cell, turbidity of cells. If adequate water is not present, it results in closure of stomata, closure of stomata. Okay, you have special cells called guard cells. These guard cells, when they are turgid, full of water, turgid means full of water, this stomata opens. Stomata opens. When the guard cells are turgid. When the guard cells are flaccid, guard cells are flaccid means guard cells do not have water. Okay, then stomata will close. Stomata will close. So it maintains the turbidity of the cell. This water is essential for growth and development. It maintains fluidity of the cell. Fluidity of the cells. This fluid media is required for enzymes for their action. Enzymes are active in fluid media. Okay, I will stop here. I'll continue in the next class. Few more external factors are there that I will take up and I will start the respiration topic in the next class.